desirable properties of plasmids as cloning vehicles. In gene cloning procedure, when we are going to use plasmids as a cloning vectors, then it must contain some suitable traits. For example, it must have low molecular weight. Secondly, ability to confer selectable phenotypes on the recipient cells. And third one, there must be a single site for different type of restriction endonucleases. Now we can focus on all these three traits one by one. So plasmid must have low molecular weight. इसका एडवांटेज ये है कि जब हम होस्ट ऑर्गेनिज्म से या रिसिपिएंट सेल से प्लाज्मिड को आइसोलेट करते हैं, तो अगर तो इसका मोलेकुलर वेट हाई होगा तो शीयरिंग फोर्सेस दैट मे बी अप्लाइड ड्यूरिंग लाइसिस स्टेप, तो हाई मोलेकुलर वेट प्लाज्मिड की डिसइंटीग्रेशन हो जाएगी. बट ऑन द अदर हैंड, if the plasmid it is having low molecular weight, its handling, its isolation, it is relatively easy. It remains stable during the lysis step. Secondly, low molecular weight plasmids, they are mainly present as high copy number plasmids. Their number per cell it is high, so that we can clone number of target DNA by using high copy number plasmid. And finally, if plasmid it is of low molecular weight, then there are chances that single site is present for different type of restriction enzymes. That is, if low molecular weight is there, then multiple substrates sites will not available. Then the next trait, that is ability to confer readily selectable phenotypic traits. जब हम फॉरेन डीएनए प्लाज्मिड में इंसर्ट करते हैं तो हमारे पास कोई वे होना चाहिए, कोई तरीका होना चाहिए कि हमें पता चले कि हमारा टारगेट डीएनए जो है वो प्लाज्मिड में इंसर्ट हो चुका है. Because there are some circumstances when we cut the plasmid DNA, it can recircularize without the insertion of our target or foreign DNA. So how it is possible to detect that our target DNA it has been inserted? So one of the way is that the plasmid must contain some selectable markers or phenotypes, so that we may have an idea that our foreign DNA it is inserted. We discussed previously that plasmids they can harbor different type of traits like antibiotic resistance, antibiotic production, metal resistance, and so on. And all these traits, although they are not dispensable for the cell, not required for the survivability of the cell, but when they are present, it can provide the cell a selective advantage over other microbes. So one of the trait like antibiotic resistance, it is frequently used as a selectable marker in gene cloning procedures. Like in some plasmids, ampicillin resistance gene is there, tetracycline resistance is there, and even chloramphenicol or other type of antibiotic resistance markers, they can be encoded by the plasmids. Then the third trait that is mainly related to the presence of single restriction sites for different type of restriction endonucleases. Suppose if plasmid contain more than one restriction sites for a single endonuclease, then what will happen? You have the idea that it will cut the plasmid DNA many times so that you can get more than one fragments of the plasmid. That is not suitable for gene cloning procedures. So, for this reason, for different type of restriction enzyme, there must be a one restriction site so that the plasmid can be converted into a linear molecule that can subsequently ligate with target DNA. And if this restriction site it is present within a gene that is readily selectable, it is more advantageous. Suppose we have a tetracycline resistance gene, and if the target site is present within this gene, then the foreign DNA can be inserted here, and after insertion, the function of this tetracycline resistance gene it is disrupted. It is no more expressing, so that cell will become sensitive to tetracycline, and this sensitivity will indicate that the foreign DNA has been inserted in the plasmid. So this process where the function of the gene it become inactivated by the insertion of the foreign DNA, it is called as insertional inactivation. And this insertional inactivation, it is a good trait for the selection of recombinant molecules after introducing it into a recipient cell. But this insertional inactivation, it is not required when the foreign DNA and the plasmid it is ligated by using homopolymeric tailing. And in other cases where the foreign DNA it also encodes a selectable phenotype.